I think yes. Okay, well, what I think is most special about being here today, I didn't know it was Dr. Lindbergh's birthday, and I feel like that's like a big deal to like give a talk on your friend's birthday. Uh, so like Dr. Lindbergh said, I am at the Arkansas Children's Nutrition Center, and I was going to give you guys just a little bit of an introduction about where I work because it is a unique place and we are a different model than most people are familiar with. Um, and so also like I live just south of you guys, but I think Arkansas doesn't always get the best press. And so I think that when the rest of the world thinks about Arkansas, these are some of the things that like Bill Clinton, one of our former presidents, he used to be the governor of Arkansas and he's from Hope, Arkansas. Um, Arkansas is the land of Walmart. So the Walton family is from Bentonville, Arkansas. We eat a lot of fried food. We have a lot of tornadoes. So last April, we actually had a really huge tornado come through the middle of town, less than a mile from my house. It was kind of scary. And also the Duggars, which I think maybe they had 20 kids, I'm not sure. But so this is, we're in the news a lot and it's never for good things. But I moved to Arkansas back in like late 2006 and I thought I was going to be there for 2 years. It's been almost 17 years and I actually kind of enjoy living there. Some of it's work, but some of it's also I do a lot of other things outside of work in Arkansas. So Dr. Siegel here might recognize this picture of this motorcycle that I used to own. We have beautiful roads for riding motorcycles. I really like to mountain bike and rock climb, and there's a lot of people that are now coming to our state for tourism, and so that's kind of frustrating. But also, it's a great thing is that we're getting better press, and occasionally we get snow. And I have dogs, and it's just there's great hiking all around the state. So these are the things that I do that are wonderful in the state of Arkansas, and actually, there's a lot of Missouri tourists that come down and hang out. So where do I work? I work at this Arkansas Children's Nutrition Center, and as Dr. Lindbergh mentioned, we are one of 6 USDA-funded research centers. They are located in the other states that are colored here in this map. And what's really unique about our center is that we're one of 2 pediatric human nutrition centers. And so the USDA, we're under this federal program that's called the National Program 107. And so typically when people think USDA, they think farms and agriculture. But the USDA has a large human nutrition research component. And so some of it is relating agriculture to food, but most of the science that we do is more related to these 4 program components 3, 4, and 5. So one of our senior faculty is on the national board for determining the dietary guidelines. And I do a lot more research here in the prevention of obesity and obesity-related diseases and life-stage nutrition and metabolism. And so another thing that's unique about our center, which can also kind of be a hindrance, is that we're a cooperative center. So my lab is actually on the campus of Arkansas Children's Hospital. And this is the only children's hospital in the area. And we serve a really large region in the Delta. So like most of Arkansas, maybe some of Missouri, and also like Louisiana and also parts of Memphis are all served through Arkansas Children's. My faculty appointment is through the University Medical Center, which is UAMS. And so we have this cooperative agreement between these 3 really large entities, UAMS, Arkansas Children's, and UAMS, which can sometimes be a great thing as we have a lot of resources, but also there's a lot of bureaucracy that comes with that. But there are also good things because of that. And so our center has a mission, which is to conduct innovative research to understand how parent-child diet, nutrition, and physical activity optimize development and maximize health across the lifespan. And so we're trying to enhance parental and child health through the science of food, nutrition, and physical activity. So just a caveat here, I was not trained as a nutritionist. I'm trained as a vascular biologist. But there's space for me here in the center to do the sort of basic science research that I'm really excited about. And just to like plug our center, like I said, we're one of 2 centers that specialize in pediatrics and also this interplay between pediatric and maternal nutrition and metabolic health. And so we have just some really cool resources at our center. So like our clinical research core, we have an inpatient and an outpatient unit. We can measure body composition like many of the labs here can do. And we also have this human sample laboratory where we can process a number of different types of human samples. Really exciting is our physical activity and metabolism core. And what's exciting about this is we're like one of the few places that has a pediatric exercise test lab. And this is run by Dr. Elizabeth Borsheim, where they can do VO2 max testing and a bunch of other things that's specifically with equipment designed for kids. We also have a pretty advanced metabolomics and analytical chemistry core, and that's growing, where we can do a bunch of untargeted, targeted lipidomics and a number of other analyses. The core that I use the most is our rodent metabolic and behavioral phenotyping core. And what's really exciting here is we have two 16-cage Prometheon systems. So these are to look at metabolic analyses in rodents. And so we can run 32 animals at a time. We can change the temperature of these cabinets, and we also have stable gas analyzers. So we can look at different, we can give the animals stable isotopes and do some assays with that. And we also have some brain research cores where we can look at brain function in kids through EEG and also through imaging. So we have quite a few resources, and this is mainly through the partnership with USDA, is that they fund a lot of this. So we have to plug our funding source quite a bit. And so just really quickly, I want to introduce my team and my collaborators. My lab is mainly run through my research associate, Liz. She does basically everything in the lab. And I have, well, he was a postdoc, but he's now been promoted to a staff scientist, Adam Corkin, who is really the brains in the lab. And then I have a bunch of great collaborators, Tiff Weinkoff in microbiology. We're doing a bunch of flow cytometry analysis, looking at platelets and how they might be involved in inflammation and perivascular adipose tissue. I have some colleagues up at the University of Arkansas. So that's 3 miles north in Fayetteville, where we're looking at venous valve devices. And then I think some of you here might know Kartik Shankar, who used to be at our center, but he's 3 or 4 years ago, he moved to Colorado. And we still do a lot of work in the programming field with the placenta. And sometimes we also have fun. So one thing I really liked about Missouri, and what I really like about universities like this, is you have a great cadre of undergraduate students and graduate students to work with. And so we really only get undergraduate students in the summer. And so when we're there, we try and have a good time and teach them a lot of things. Inevitably, they all go to medical school, but I think that as long as some of them learn a little bit about research, then maybe I've accomplished something. And yeah, we took this silly picture at the new APS Summit last year, Adam and I. And so I'm pretty excited to attend that meeting, and hopefully we'll see a bunch of you there next spring. So I'll just really briefly give you a broad overview. So I do a lot of different things in my research, but my research is really focused on cardiovascular health and vascular biology. And I'll just give you a brief overview of some of the projects we're working on, and then we'll talk a little bit more specifically about maternal programming of perivascular adipose tissue. So the main focus of our USDA research is understanding how maternal obesity in both humans and animals can program cardiovascular disease in offspring. And so the research I'll tell you a little bit about today is in our preclinical models and our mouse models, how maternal high-fat diet can program perivascular adipose tissue dysfunction. But alongside that, we have a number of clinical cohorts at our center, and we have these longitudinal studies understanding how maternal obesity during pregnancy can program health and disease in kids. And when they started this study, they weren't measuring any cardiovascular outcomes. And I was like, oh, this is a great opportunity. I can bring something to this lab. And so I was really interested in measuring endothelial function in kids from this cohort. So their mothers were either normal weight or they had overweight or obesity early on in pregnancy. And we followed them and from birth, and now they're turning 8 and 11 years of age. And so we've been measuring endothelial function using flow-mediated dilation. I know that's a technique that many people here are familiar with. And so, because there really isn't much in the literature about how early do we see endothelial dysfunction in the pediatric population, and is there an association between maternal health and early cardiovascular dysfunction in kids? I have an interest in the integrated stress response pathway and how that might be involved in perivascular adipose tissue function. And I'll talk a little bit more about this in my talk. My postdoc, Adam, is actually a platelet expert, and platelets are important in bringing immune cells into tissues. And so he has this idea of maybe platelets are important in bringing leukocytes and other immune cells into perivascular adipose tissue under stress conditions. And so he has a small study to look at that. And then with my collaborators at the University of Arkansas, we're working on developing a prosthetic venous valve device, because at one point in my life, I studied veins and I was just focused all on venous physiology. So it's been really neat to see how some projects, so some things that were things that I studied in graduate school and my early postdoc, while I'm not totally focused on them now, I'm still being able to bring those back into my research career. And this is the first time that I've been involved in like a device development sort of grant, and this is really new and exciting. But we're still working on getting more funding to do this. And so that's also been an experience of going through NSF, which is a completely different beast than NIH. And aren't grants fun? And so if we talk about our USDA funded research, we know that obesity is increasing in the US. And basically in this animation, what you're seeing is that the prevalence of self-reported obesity is increasing over time. And so the darker red colors here are states that have extremely high obesity rates in adults. And my home state of Arkansas, like greater than 35% of the adults are classified to have obesity. And since we study pediatrics, if we look at the pediatric population, the darker blue colors over time, and this is from 2003 to 2017, it's the same thing. We see this increased prevalence of obesity. This is among high school students, but this is also occurring at younger and younger ages. And so in Arkansas, approximately 22% of high school age students have obesity. And this is in 2017. So this is even old data, and it's not any better today. And so this is a problem because we know that obesity is associated with health outcomes later on in life. And so now we're seeing children that have sooner prevalence of what we previously thought were adult-related diseases. And so the interest in understanding maternal programming is that we know that nearly 60% of women who become pregnant have overweight or obesity. So that's a very large percentage of our population. And since we know there's a high percentage of childhood obesity, what is the relationship between maternal obesity and childhood obesity? And so this leads us to the fetal origins of health and disease. And so this is a magazine article from Time from like over 10 years ago. And the title here is How the First 9 Months Shape the Rest of Your Life. And so this is an acknowledgement that there are critical periods of development that are important in determining our health later on in life. And this hypothesis was first put out there by Dr. David Barker. He studied some populations in England this is shortly after World War II, and also in the Netherlands. And he observed that there were these cohorts of women that had undergone nutritional deficiencies, mostly protein deficiency, during certain times of pregnancy. And when he studied their offspring later on in life, like at age 40, there was an increased incidence of cardiovascular disease. And so he hypothesized that the nutritional status during different stages of pregnancy can lead to the increase of risk of cardiovascular disease, hypertension, diabetes, and obesity. And so we now have several societies studying the developmental programming of health and disease. There's a US DOHAD, which is Developmental Origins of Health and Disease. And so it's recognizing that we have fetal programming during critical windows of fetal development, but also there's postnatal programming. So there are other windows of development besides fetal development, such as lactation and also early childhood, that when we have different environmental insults in here, these may program disease later on in life. And also the question is, since we know that there's these critical windows of programming, can we prevent the development of these adult diseases by intervening or having some interventions either during fetal development or later on in postnatal development? And also, there is just different susceptibility to obesity and other metabolic alterations because of these developmental origins of health and disease. And so when we are talking about this developmental programming, we think about the first hit occurring really early on in development, so typically during fetal development. And this first hit can be environmental, and the environment can, like if we think of the in utero environment, it could be maternal diet, it could be external factors such as temperature, like heat stress. It could also be maternal stress from whatever source. And this has been shown to cause organ-specific metabolic programming. And there's a number of organs that can be affected. These depend on the type of environmental stress, but the pancreas is definitely a target organ, as is the liver, adipose tissue, and also gut development. And then later in life, we can have a second hit. And so again, in our studies, we use diet, again, as the second hit, but it could be lack of physical activity or other sorts of things. And this, in the face of this first hit of organ-specific remodeling, can cause additional metabolic remodeling and dysfunction. And so then you can see increased prevalence of diseases like non-alcoholic fatty liver disease, adipose tissue dysfunction, and gut dysbiosis, and then perhaps early metabolic and cardiovascular disease. And so I like this picture here because they put a picture of a blood vessel, and as I mentioned, I'm a vascular biologist. But when I look at this picture of the blood vessel, something bothers me a little bit. And so this is the classical picture of blood vessel anatomy that we learned, that there are 3 layers to the blood vessel. The innermost layers are endothelial cells, our smooth muscle layers are in the intima or in the middle, and we have an adventitia outside of the smooth muscle cell layer. But again, something's missing from this picture, and this is actually not true for most of our peripheral vessels. And that thing that's missing is the perivascular adipose tissue. And so we can liken this to being the 4th layer of the blood vessel. And so most of our peripheral vessels have a layer of perivascular adipose tissue that surrounds the perivascular nerve network and the adventitial layer of the blood vessels. And interestingly, so a number of like Michelangelo and some other medical illustrationists dating back hundreds and hundreds of years, when they drew the human anatomy, they actually omitted this layer of fat. And so I think it wasn't really discussed because most of the medical illustrations didn't have this layer depicted there. And it has important physiological functions. I'm not sure why they didn't include it in their drawings, other than maybe it was more unsightly than like a muscle or something like that. But there's an interesting historical perspective on why perivascular adipose tissue was not studied for a really long time. And so one thing that's interesting about perivascular adipose tissue is that the origins and adipocyte composition of perivascular adipose tissue really varies throughout the length of the body. And so the progenitor cells are different in different regions. So this is a picture of the aorta. And so it starts off with the aortic arch, and then below the diaphragm, and then branches to the abdominal aorta. And so the progenitor cells that are present in the thoracic aorta are different from the progenitor cells in the abdominal aorta, and also the perivascular adipose tissue that is on the mesenteric arteries. And that is important because the adipocyte composition is different. So we know that there are different types of adipose tissue. There's white and brown adipose tissue that has different functions and different metabolic functions. And also, in general, in the thoracic cavity, most of the perivascular adipose tissue is more brown-like. In the abdominal aortic cavity, it's a mix of white and brown, so they like to call that beige. And then in the mesenteric arteries, has a more white-like phenotype. And so perivascular adipose tissue has a function. It is not just surrounding the blood vessel and causing, or some people originally they thought it was just structural and it was holding the blood vessel together and preventing it from bursting. But it actually releases factors. It releases what they like to. So there's endothelial derived relaxing factor, but there's also adipose derived relaxing factor. And so in these really early studies, they just did some transfer of the physiological salt solution in these organ bath chambers, where in the acceptor tissue, they had just dissected the adipose tissue off the aorta they were studying. And in this donor organ bath chamber, the perivascular adipose tissue was still attached to the blood vessel. And so they then just transferred the solution, had the factors that the perivascular adipose tissue released, and they saw that there were changes in constriction. And so what this looked like is, if we look at here, just so angiotensin 2 is a really potent vasoconstrictor. And so if we look at this graph here in A, they're adding increasing concentrations of angiotensin 2, and we have the force or tension on the y-axis, and it says they add increasing amounts of angiotensin 2, the blood vessel constricts. So there's more force being generated. And so this top panel in A, they have removed the fat off. This is what I did all throughout my graduate studies. It's really easy to cut it off the tissue, which is kind of odd, but it's easy to study for that reason. But when they left the tissue on the aorta and they added angiotensin 2, the constriction of angiotensin 2 was completely gone. And so they said, oh, I guess the perivascular adipose tissue is releasing a factor that's preventing vasoconstriction. Because if we take it off, then we now see vasoconstriction. And we can, so this is on C is just summarizing the data from the graph here in A. And this is true for other vasoconstrictors. So angiotensin 2 binds to AT1 receptors, could cause vasoconstriction, but there are other active molecules like serotonin and phenylephrine that induce vasoconstriction, but they bind to different receptors. So this wasn't just a phenomenon of angiotensin 2. And so further studies have characterized PVAT a little bit more. And like I mentioned, there's an anatomical difference in the adipocyte composition and phenotype. So our mesenteric arteries have typically more white adipose-like perivascular adipose tissue, whereas the thoracic aorta has more of a brown-like phenotype. And there's a number of adipokines and cytokines that perivascular adipose tissue can secrete, and that these can all have effects on vascular function. And also, it's important in modulating insulin signaling. And so one thing that's really interesting about perivascular adipose tissue is it can be modulated based on with diet and exercise, and how, so it's a factor that can be modulated. And this is where I think it can be translated into clinical studies, is that while I'm studying animals, there are interventions that we can do in animals to affect perivascular adipose tissue function. And so I think we can make that similar correlation to the human condition. And so under normal conditions, perivascular adipose tissue is anti-contractile. So it's normally trying to keep the blood vessel open and dilated, keeping our blood, reducing total peripheral resistance, and keeping our blood pressure low. And it releases a number of anti-inflammatory factors, anti-proliferative factors. And so the general thinking is that this is protective against cardiovascular disease, be it atherosclerosis or hypertension. But if you perturb the system, like other systems, it can become dysfunctional. And when it becomes dysfunctional, it loses its anti-contractile properties. And it does that through impairing insulin signaling, increases cytokines that are produced, it increases reactive oxygen species production, it affects nitric oxide bioavailability, and it also changes the composition of adipokines that are released. And so it leads to inflammation, hypoxia, oxidative stress, insulin resistance, and just in general vascular dysfunction. So an important feature of perivascular adipose tissue, and that while other adipose tissues can change with different perturbations like obesity, this has a direct effect on vascular tone and vascular function. And related to my research, there are a number of studies out there showing that prenatal exposures can alter perivascular adipose tissue function. So this is proof of principle that factors during these critical development periods can affect PVAT function. And so in this one study here, so these are a little bit older, like about 10 years old, they exposed rodents to nicotine prenatally, and then looked at the function of perivascular adipose tissue using phenylephrine as a vasoconstrictor. And so in this panel C, they have the fat still attached to the aorta, and in panel F, they've removed the perivascular adipose tissue. And so then the nicotine and saline are the pups or the offspring that were either prenatally exposed to nicotine or saline. And you can see that when they were in the presence of perivascular adipose tissue, there was a greater constriction in the nicotine exposed offspring. And if we remove the perivascular adipose tissue, that response is lost. And also, in line with what I'm interested in studying, this was an experiment done in rats where the female rats were made obese through diet. So they were given a high-fat diet for 12 weeks, and then their male offspring were studied. And so this panel C is the control diet, and panel D is the moms that were fed a high-fat diet. And when they looked at the effect of perivascular adipose tissue on constriction, so this is just another vasoconstrictor that binds to thromboxane receptors. In the control condition, and we can see that perivascular adipose tissue has an anti-contractile effect. So in these black or the dark circles, this is the absence of perivascular adipose tissue, so it's a greater constriction. And then in the open circles, this is the presence of perivascular adipose tissue, there's a reduction in constriction. And so these are the offspring that are from moms who were fed a high fat diet. And when we looked at offspring, sorry, that was control. And when we looked at offspring who were from moms that fed a high fat diet, this perivascular adipose tissue lost its anti-contractile properties. So the gray or open circles, in fact, probably have greater constriction to U46619. So this is proof of principle that dietary factors and also environmental factors during pregnancy can actually program perivascular adipose tissue function. And so that was where we were at. So to summarize, perivascular adipose tissue is important in modulating vascular activity through secreting adipokines, cytokines, and reactive oxygen species. But the molecular and epigenomic basis of PVAC function are still poorly described. And how perivascular adipose tissue responds to alterations in the in-utero environment are still not fully understood. There's a few studies out there, but there really hasn't been a deep characterization of the effect of the maternal environment on offspring PVAC function. And so we developed a mouse model to answer the question of, does maternal diet in utero program offspring perivascular adipose tissue and arterial function? And so in this mouse model, we made our female mice, or we fed them a high fat, high sucrose diet for 12 weeks before we bred them with lean male mice. And then when their pups were born at weaning, we randomized their pups to either a control diet or the same high fat, high sucrose diet that we fed their moms. So we have 4 groups of offspring. And I'm going to use the same like naming conditions here, where the first letter refers to the maternal diet. So C is for control diet and H is for high fat diet. And then the second letter refers to offspring diet, or the offspring post-weaning diet. So again, control and high fat. And so we have our 4 groups, and we also have collected tissues from both male and female offspring to see if there are sex differences. I'm not going to show you data from all the males and females, because in these studies we ended up generating a lot of animals and a lot of data. And if any of you have done contractility studies, I don't have quite the machine of I have a myograph, which is 4 channels, but if I had 8 channels, then we could do more mice and just crank through a bunch of mice all at one time. But we also measure a bunch of outcome in the offspring. So while my main interests are in arterial activity and also gene expression in the perivascular adipose tissue and the corresponding arteries, we have our whole rodent metabolic phenotyping course. So we do have detailed data on body composition, indirect calorimetry, and also glucose disposal. But I'm not going to show you the GTT data just because there's a lot of it. So the first thing we saw, though, was that this maternal high-fat diet before pregnancy can program body weight and body composition. And so this is in the male offspring and female offspring body weight over time. And so we have our 4 groups here, where the purple is the pups that were exposed maternal high-fat diet, and then they were fed a post-weaning high-fat diet. And then the pink line is our control animals. So the moms are fed a control diet, and then they were also fed a post-weaning control diet. And what we'd noticed before in different rat models looking at maternal high-fat diet and obesity was that it was really this combination of maternal high-fat diet and offspring high-fat diet challenge that programmed this excessive weight gain. And we see this in both our male and female cohorts, though it's more pronounced in the males. If you look at the scale here, the males in general just gain way more weight. But again, it's this combination of maternal high-fat diet is causing excessive weight gain in the face of an offspring high-fat diet post-weaning. And then we have echo MRI. We just got a DEXA machine for the mice, but we haven't put them in there yet. So this is a little bit older study, but we can look at fat mass and lean mass in these animals. And so we're trying to think of the best way to show this, because if you just look at the numbers, it looks like there's huge changes. But so we were looking at the percent change in these different compartments, and the males, like the offspring high-fat diet, has a really strong effect on fat mass gain. And it really seems like they're gaining more fat mass with the maternal high-fat diet and offspring high-fat diet. And percentage-wise, they're not gaining lean mass. And so their weight seems to be more, and this actually seems to hold true in the females. And so this is kind of unique in this model, is that we do see some programming of excessive weight gain and adipose tissue gain in our models in both males and females, which has not always been shown in literature. And then, like I mentioned, we have these two Promethean systems to look at indirect calorimetry. And so one of the questions we want to ask is, are there gross changes in energy expenditure that could be explaining these changes in body weight and body composition? And so we can split up the measurements of when we're measuring metabolic parameters during the light phase and the dark phase. So are the animals active or are they resting? And we can look at factors like respiratory exchange ratio and energy expenditure. And so this is actually just in the male offspring, because there's a lot of data to go through this. And so not surprisingly, we see with respiratory exchange ratio that the offspring that were fed a high-fat diet had a lower RER. And this really is indicative of their diet's different, right? There's more fat in there. And so this is telling us really that this is the appropriate response, that our machine is probably working properly, that we see a change in the substrate source. And so we see a change in RER. And we also see an increase in energy expenditure, and especially in the group that was from the yams were fed a high-fat diet, and also a post-weighing high-fat diet. But one thing it's important to note is that these animals were also heavier. So when we did ANCOVA and you use body weight as the covariate, then body weight, actually the covariate was significant. And so really this change in energy expenditure, we think, is actually being driven more by body weight than actually maternal programming. And I think that if you plot like these percent frequency response curves for RER here on the top and energy expenditure on the bottom, you can clearly see for RER, there's a strong diet effect. And that's because the substrate is different, right? The animals are fed a high-fat diet. They're obviously going to have a difference in energy utilization. And then in our animals, looking at total energy expenditure, really body weight is the confounder here. And so one thing we're thinking in these studies, all of these studies were done at 21C. And so these animals are pretty cold. And so they're having to activate their brown adipose tissue to stay warm. And so one of the things that we're doing in this next round of our project plans is actually doing experiments at thermoneutrality. So under these conditions, like these animals all have quite a bit of brown fat activation. And so the energy expenditure might be confounded by the fact that they're spending a lot to stay warm. Because actually when they're in the Promethean, they're singly housed. They're given some bedding and material, but they really can't cuddle with each other. And so they're probably actually kind of cold the whole time. And so we're doing some of these studies at thermoneutrality to try and remove this confounder of, and I'm kind of curious to see if we're masking some of the effects of maternal high-fat diet because the animals are spending, the offspring are spending so much energy trying to stay warm. And we have my colleague Craig Porter has some preliminary data showing that there really are profound changes in energy expenditure by changing the temperature from 21C to 27C just for a short period of time. And so just it's a kind of a thing that I don't think it negates any of the data that we've collected in the past, but if we're trying to build models to more properly mimic the human condition, maybe we need to rethink about how we actually do these studies a little bit. A little frustrating, but it depends on which strain of animal you're studying, but it can vary, and the range is, I think, from 27 to 29, depending on the strain of mouse. And I think maybe in rats, it might be a little bit different. And so it's also, so the Promethean is really cool because we have these cabinets and we can shut the door and you can maintain the temperature pretty stably. But we've also been doing some experiments where we have a small room in our animal core and they're just increasing the temperature in the room, which was great until it was winter and we had a pipe burst, and then they had to turn the heat off in the building during like the coldest day of the year. And I was like, dude, this is in the middle of my study. Like, this is not okay. But the backup plan, we put them in the Promethean. So there actually are environmental challenges to actually running studies at thermoneutrality because people are not comfortable, right? They don't want to go in there to change cages. But that's the plan of trying to now do all of our rodent studies at thermoneutrality for a mouse to more. But then the question is, if we pull them out to do a glucose tolerance test, the room that they're in is 21. Pardon? Yeah, exactly. And so we collect the brown fat adipose tissue, but again, maybe that depot is artificially larger because they've been housed under cold conditions. And so really the plan is to compare our thermal neutrality studies with the cold condition and pair that with the energy expenditure measurements to get a better picture of what actually is happening in these animals. So you get new equipment and you think you can make a cool story and tell more studies, but then you just have more questions. And like, oh, maybe we didn't do this perfect the first time. But maybe that's also science. And so then we also, so after we've done all of these in vivo studies in the animals, I was interested in contractility of the vessels. I started out with the thoracic aorta because the mouse is tiny and their vessels are not very big. And so we used this U46619, this thromboxane receptor activator, to look at contractility of the thoracic aorta. And we looked at it in the presence and absence of perivascular adipose tissue. And so the open boxes here are the tissue with the PVAT still attached. And then the filled black squares are with the PVAT present. And so with increasing concentrations along the x-axis of our vasoconstrictor, we see increasing contraction. And in the presence of PVAT, this contraction is reduced. So at least in our control animals, we see that in our hands, the perivascular adipose tissue is indeed anti-contractile. But when I look through all of our groups, even, so these are the animals from, lambs were fed a high-fat diet, and then the pups were fed a control diet. And in this case, also perivascular adipose tissue was anti-contractile. And then if we looked actually at our last group, so in all of our conditions, even with the high-fat challenge post-weaning, our perivascular adipose tissue was still, in the presence of it, was still anti-contractile. And so that was a little bit disheartening in that I thought perhaps we didn't, like, were they not challenged with a high enough fat in their diet, or like I thought there should be some more perivascular adipose tissue dysfunction. But if we did compare just the curves to U46619 from our control animals and then the animals whose moms were fed a high-fat diet and post-weaning a high-fat diet, and we looked at the EC50s, so this is the concentration of a drug that causes 50% contraction, we do see that there is a leftward shift in the thoracic aorta. So that's something that's changing. Yeah, the age was the same. We have sections of them, the tissue, so we could actually go, and something that we need to do. I have so many histology tissues that are waiting to be analyzed, but that's a good point. That specific tension. Yeah, it's a great point. And so this is in the absence of perivascular adipose tissue. So in the thoracic aorta, there didn't really seem to be too much of a maternal programming effect with this high-fat diet. But in the aorta itself, perhaps it could be a size issue, but there is some inherent differences in arterial function. But also, so if any of you know anything about blood pressure, if you've been in the blood pressure world, the thoracic aorta is not the tissue to study because it is not a resistance vessel. And so we have a wire myograph where we can study much smaller vessels like the mesenteric arteries. So these are the second order mesenteric arteries that are more important in blood pressure regulation. And so these studies, we looked at contraction to norepinephrine, another vasoconstrictor. And then the black bars is the constriction where the PVAT has been removed. And the gray bars is where the PVAT is still attached. And then we have our 4 different groups of offspring. So our control fed dams that were fed, and the pups are fed a control diet. And then on the far right, your left, wait, you're right. I'm sorry, I get confused left and right. But this is the combination of maternal high-fat diet and offspring high-fat diet. And so we can see that in this case, our perivascular adipose tissue, again, is anti-contractile. But in our HH group, we're losing the anti-contractile effect of perivascular adipose tissue. And so perhaps because there's a different phenotype of the perivascular adipose tissue in the mesenteric bed than the thoracic aorta, we're seeing here that the combination of maternal obesity and offspring high-fat diet is actually affecting perivascular adipose tissue function. And this is also true for a different vasoconstrictor like angiotensin 2. Maybe there's a trend for a decrease in contraction in the HH group, but this was not statistically significant. And so I think that perhaps the mesenteric bed is a better vessel area to study to look at maternal programming of perivascular adipose tissue function. And these are more functional as far as blood pressure regulation. And so, like I mentioned earlier, that perivascular adipose tissue can release a number of molecules that regulate vascular tone. And this is from a review paper that lists a number of these factors that are released. One really important vasodilatory factor that is released from perivascular adipose tissue is hydrogen sulfide, or H2S. And hydrogen sulfide is synthesized through two enzymes, cystathionine beta synthase and cystathionine gamma lyase. I don't know why they call it CSE, but that's what they call it. And this is a really important vasodilator that acts by opening potassium channels in smooth muscle cells. So it can diffuse from the PVAT into the smooth muscle cells and cause vasodilation. And so I was curious, it's kind of hard to measure H2S because this is a volatile chemical, but we can measure the protein or the gene expression of these H2S synthesizing genes. And what we can also do is we can use drugs to block these enzymes and decrease H2S production. And so there is, this is a very complicated pathway of hydrogen sulfide production, but we can use this drug called propargyl glycine, and it blocks a couple of different steps. It blocks both these enzymes that are involved in the production of hydrogen sulfide. And so that's what we did in our contractility studies. And so this is the contraction to norepinephrine in the presence and absence. So presence of PPG in the gray bars and the absence of PPG in the black bars. And so this is in the absence of perivascular adipose tissue. And basically you can see that the blocking H2S production really had very little effect on norepinephrine induced contraction in all of our 4 groups of offspring. And then in the presence of PVAT, and so like this N is pretty small, it's only an N of 6, but you can start to see that in the presence of PPG, at least in our control animals, the contraction in norepinephrine is a little bit larger. And this holds true for most of the groups of offspring. However, when we look at the HH offspring, so the ones that were exposed to maternal high-fat diet and they were fed a postnatal high-fat diet, PPG has seemed to lose its effect. So we think that there's perhaps a change in hydrogen sulfide production in the perivascular adipose tissue. And this has been demonstrated actually in other tissues that obesity is associated with a decrease in hydrogen sulfide production, but it really hasn't been in the perivascular adipose tissue field that's been demonstrated. And so this was kind of some interesting and exciting data that we generated. So we then looked at gene expression of these enzymes that are important in producing hydrogen sulfide in the perivascular adipose tissue. And it seems like there really is a strong diet effect on the expression of these 2 hydrogen sulfide producing genes. I was hoping that there would be a change in these enzymes in just the group that was exposed to maternal high-fat diet alone, but also offspring high-fat diet is a really strong modulator. And also these animals, like I said, these studies were done at they were done when the animals were cold. But I do think that this is promising because it is recapitulating what other people have published, that a high-fat diet can modulate the expression of these hydrogen sulfide producing enzymes. And this is doing this in the perivascular adipose tissue. And another thing that why I'm interested in this is that so there's this pathway called the integrated stress response pathway, and this is activated during severe protein restriction. And so typically when someone has severe protein dietary restriction, you don't want to make a lot more proteins. And so your body shuts down a lot of the enzymes that make proteins and make muscles. But there are some things that are activated by this response. And what's activated is called the integrated stress response pathway. And one way we can mimic this is through decreasing dietary methionine and cysteine in rodents. This is also called a vegan diet, but there's some things like soy that have a lot of dietary methionine. But it basically increases expression of this transcription factor called ATF4. And ATF4 can increase the constriction of a number of genes. And so Tracy Anthony, who's at Rutgers, was giving this talk. And so this gene right here that they called CTH, this is also cystathionine gamma lyase. And she was saying, oh, this is one of the proteins that is upregulated by dietary methionine and cysteine regulation. So I was like, oh my gosh, did you measure blood pressure in these animals? And she's like, oh no, we were just really interested in the liver function and skeletal muscle function. And so I have a small study looking at dietary methionine restriction and perivascular adipose tissue function. And so that's tying in how, since this is the protein that's involved in hydrogen sulfide production, this is why I thought this is a factor that could be modulated by maternal high-fat diet and offspring high-fat diet in perivascular adipose tissue. And so when we look at some of these genes in this pathway, PERK is actually upstream of ATF4, or it's over here, that is activated, or is down, sorry. I'm getting these all backwards, but it's involved in the integrated stress response pathway. And we do see decreased gene expression of PERK, at least in the animals that were exposed to maternal high-fat diet. And there doesn't seem to be a very strong offspring high-fat diet effect here. And perhaps there's some effect of GCN2. But we were most excited about the changes in cystathionine gamma lyase and cystathionine beta synthase in our maternal high-fat diet feeding studies. And so, like I said, this is actually a rat study that I'm doing where we think that under normal conditions, since perivascular adipose tissue makes hydrogen sulfide, which is important in vasodilation, we think that perhaps with dietary methionine restriction, this will activate the integrated stress response pathway, increase ATF4, and potentially increase cystathionine gamma lyase expression. And because obesity is associated with this decreased hydrogen sulfide production, perhaps this dietary methionine restriction would be a way to restore that vasodilation and perivascular adipose tissue in obesity. So this is like actually like a little bit of an aside. It's a rat study that I think is still related to the whole study of perivascular adipose tissue and maternal programming, but a little bit of an aside. And so the next thing we did was use RNA-seq to look at gene expression of perivascular adipose tissue from our 4 groups of offspring. And so we use RNA-seq, and this is just from the male offspring, and this is just from the thoracic PVAT, which I told you earlier, didn't really have much of a functional effect. But one thing I thought was kind of interesting, so this is differential gene expression. So these are the genes that are differentially expressed between these different groups of animals. And we looked at the PVAT, we saw that if we just looked at the effect of maternal diet alone, there were only 29 genes that were differentially expressed at a fold change of 2 or higher. If we looked at the effect of offspring diet in perivascular adipose tissue, there were quite a few more genes that were affected by offspring high-fat diet. But when we looked at the combined effect of maternal obesity and offspring high-fat diet, there were exponentially way more genes that were differentially expressed. And so there really is this combination of maternal obesity and offspring high-fat diet. So that's first hit and the second hit that are causing a significant change in gene expression in the perivascular adipose tissue. And I would guess that this is true for other tissues also. One thing that's interesting, we did this RNA-seq also in the paired aortic tissue, and maternal high-fat diet had a strong effect on gene expression in the aorta, but offspring high-fat diet didn't have nearly as strong of an effect on gene expression. I don't know the answer or the reason for that, but in the aorta also, there was a exponentially greater effect of the combined effect of maternal and offspring high-fat diet. And so when we look at the different biological processes and genes, we see a lot of important genes that are involved in lipid metabolism and lipogenesis. Geez, I can't talk right now. And there also seems to be a number of genes that are important in fatty acid metabolism. And this is all in the perivascular adipose tissue. And this is the genes that are differentially regulated by the combination of maternal high-fat diet and offspring high-fat diet. And so these are some targets that we are planning to investigate further. I don't know about you, but RNA-seq data can also be like it's a fishing expedition. There's so many targets and you want to look at all of them. And so narrowing this down to genes that are important and function has really been a little bit of a struggle for me, but we're moving ahead with that. And we've also done RNA-seq in the mesenteric vessels and the mesenteric PVAT too. And so I hope I've just imparted a few things. The early life events are really important in determining chronic disease risk. That maternal obesity is important in contributing to offspring cardiovascular disease risk. PVAT is an important modulator of obesity-associated cardiovascular disease, and that maternal obesity can program perivascular adipose tissue function and gene expression. So with that, I'd like to thank all of you for meeting with me all day long, for your attendance, and I'd be happy to answer any questions.